introduction. So pleased to introduce Paul Gignac. He's our speaker today. He's an evolutionary, I have a very nice intro from Sarah Warning. He's an evolutionary biomechanist and vertebrate paleontologist. His research focuses on how highly integrated phenotypes evolve without compromising that function. He's particularly interested in musculoskeletal and dental development and evolution among fossil and extant archosaurs, especially crocs and other reptiles. And Paul received his Bachelor of Science degree in Ecology and Evolutionary Biology in 2004 from the University of Connecticut. And his senior thesis honors advisor was Kurt Schwenk. So that's his first connection to Marvel Wake, academic grandchild. And for that thesis, he examined cranial kinesis in lizards. And then he received his PhD in biological science in 2010 from Florida State University. And his PhD advisor there was Greg Erickson. So that's his second connection to Marvel Wake. So he's a dual fork academic grandchild of Marvel Wake. And his dissertation there examined ontogenetic changes in alligator feeding biomechanics. And currently, he's an instructor and researcher in the Department of Anatomical Sciences at Stony Brook University. And today he's going to be speaking on his work with crocodile feeding biomechanics. So welcome, Paul.
All right, thank you very much. Today I'm going to talk about how crocs have managed to dominate as apex predators for the last 85 million years all over the world. And I'm going to turn my title on its head a little bit. I'm going to start off talking about the evolution and development of bite forces in crocodilians, and then pivot to biomechanical modeling as a way to address both constraint and plasticity in their jaw system. And along the way, I'm going to sprinkle in some details of a methodological advancement that I've been working on at Stony Brook to fully visualize vertebrate soft tissues using micro-CT technology. All right, so as Jim introduced me, I'm very interested in complex phenotypes. This is my legacy from Kurt Schwenk. He got me addicted to this. And so what I'm interested in is how do these highly integrated, very networked anatomical systems change over development and at cladogenic events without compromising their inherent function. And to look at this, I explore crocodilians. This is a really great group for understanding their diversity and testing questions about how they've exploited a myriad of different feeding niches. They live in their depositional environments, and as a result, we have a really great understanding of their evolutionary diversity. That diversity is mostly contained within the skull, and there's a kind of a continuum of cranial morphotypes. We've been into sort of 6 general categories. Zyphodont crocs, which have a very deep skull and blade-like teeth. The paleosuchids, which are still around today, that have a somewhat dog-like vaulted rostrum. The blunt and slender-snouted forms. Generalist or intermediate crocs, and according to Chris Brochu, the so-called duck-faced crocodilians. Now, these cranial morphotypes are considered ecomorphologies. They're thought to form a tight relationship to the organism's environment, in this case specifically assumed to relate to their ecological feeding niche. And so over their evolutionary history, we've seen that taxa that occupy similar environments have similar kinds of ecomorphologies, whereas taxa that are closely related don't tend to show those. That is, ones that are endemic to the same areas diverge quite distinctly in their cranial morphotypes. Their postcranial anatomy is highly conserved, and so because of this, we think the keys to their niche shifts have really relied on transitions in diet, and that those have then manifested as differences in their cranial morphology. Today, 4 of these ecomorphs are present among extant faunal assemblages, and these ecomorphs are related to presumably their diets. And so most, or actually all, crocodilians are opportunistic feeders, but we can generalize that generalists have a very catholic diet, whereas slender-snouted forms will eat small compliant prey, specializing on fish, on arthropods. Blunt-snouted taxa tend to be duraphagous, and those with the more vaulted rostra, the paleosuchids, exploit terrestrial prey mostly, especially small mammals. And so these morphotypes have been, for a long time, considered to reflect a number of aspects of their biology, and in biomechanics it's thought that their bite force capacities would relate to the differences in skull strength here, so that gracile forms with slender snouts should have the relatively lowest values of bite force, and that presumably limits them to the kinds of prey that they're acquiring, whereas beam theory suggests that blunt-snouted forms and those with more tall rostra should have relatively higher bite forces, and generalists will fall somewhere in the middle. Now, These ideas, in sum or in whole, go back more than 100 years, in fact. The first bite force test that was ever done on any animal, as far as I can tell, was done on a Siamese crocodile, and it was reported in the London Times of 1882. So crocodile biology has been kicking this around for a long time, and it's sort of become standard in the field that we assume these relationships are true. And it's only really been recently that we've been able to get the technological advances and access to enough specimens to test these ideas. This is a figure from McHenry et al. And his group in Australia from 2006. They used finite element analysis to explore the safety factor of snouts in a diversity of crocodilians. So slender snouted forms, blunt snouted forms, more intermediate snouts. And they found a 9-fold difference with this is the freshwater crocodile having the relatively weakest snout, and the blunt snouted forms, this is a Camelotorostrus, having a snout that's 9 times stronger in bending. And so presumably then they inferred from this that bite forces should vary by as much as 9-fold as well. But crocodilians don't just use their bite forces for acquiring prey. They're used in defense, they're used in fighting and combat, they are used for social dominance. That even includes these slender snouted forms. So getting access to mates and good feeding areas is dictated by how big an individual is and how much force it can generate, and therefore how much damage it can inflict on a potential competitor. So there's more going on here than just the feeding biology for these snouts, and that suggests that this may be somewhat limited in terms of its explanatory power. Now, ideally, we would test this by measuring bite forces in a diversity of extant crocodilians. And that would give us a really comprehensive understanding of how their feeding biomechanics change over their numerous cladogenic events. And it would once and for all address this question: is jaw shape coupled to feeding biomechanics, particularly bite forces? Well, not to be outdone by Victorian-era biologists, Greg Erickson, myself, and our colleagues went and did just that. We sampled bite forces from all 23 extant crocodilian species. Our data set includes 85 individuals. Most of these are represented by multiple individuals, and they're all adults. And if you subscribe to any of the newer studies that have suggested there are as many as 24, 25, or 26 extant species, members of those various populations from the American crocodile, the Nova Guinea croc, and the Nile crocodile are all represented in this data set. So we don't expect our results to change based on what new taxonomic crocs are named in the next few years. Okay, so we measure bite forces using a force transducer-based bite bar. This is padded with leather to protect the animal's teeth. We restrain our specimens, not me, we actually restrain them down to a board so that they don't move during biting, and that does not add any undue loads to our force transducer. So they're not able to, they're not giving us any forces beyond what their jaw muscles are generating in and of themselves. Now, we use defensive bites, or we only use defensive bites in our analysis, so bites that weren't loaded completely for their duration or were associated with some odd behavior, we didn't include. We measure our bite forces at the most prominent malariform tooth, and that sits in the apex of the second maxillary convexity. This position is fairly equidistant in all extant taxa. And even in the slender snouted forms, there's a larger tooth and a larger alveolus at this position. So it's both biomechanically and ecologically relevant as a place for us to measure these values. And so here are the results from that study. All of the cranial ecomorphs are differentiated by colors. And what I want you to really notice here is that body mass explains the vast majority of variation in interspecific bite forces. So each one of these is an average value for one taxon. You guys have a additional pointer. I just died.
We have a
stick.
You have a stick?
Old-fashioned.
I'll use the stick.
old school.
Excellent. All right, so what we found is that body mass explains the vast majority of bite forces. So these are taxon representative values, and with one exception, Gavialis gangeticus, all taxa fall within a 95% confidence interval that explains bite forces in terms of body size. And this surprised us. This essentially goes completely counter to what has been expected for the last century or so, relating cranial morphologies to feeding biomechanics. And so what it shows is that these bite forces are not coupled to jaw shape. We also examined these data in a phylogenetic context and removed the effects of phylogeny, and still body size explains 87% of the variance in bite forces. If we take that remaining 13% and account for snout morphology, that explains only 19% of the remaining variance, and in total, 2.5% of the total variance in bite forces is related to snout morphology. So we can say pretty confidently that bite forces are uncorrelated with snout form. And what this is telling us is that in terms of looking at their evolutionary history, body size is the overwhelming signal. And so we've done a number of examinations where we've reconstructed bite forces in fossil taxa, for example, the very large Deinosuchus riograndensis. It's about a 40-foot long crocodile and or alligatorid. And it generates values of around 100,000 newtons, about 25,000 pounds of bite force. So we're able to do ancestral character state reconstructions on performance by transforming body mass estimates into bite force capacity, or bite force predictions. But these are all adults, and we know from some feeding systems that taxa can take different routes to arrive at the same, different developmental routes to arrive at the same adult phenotype. And so it's possible in these animals that ontogeny is playing an important role. And the reason we may think that is that there's a 5,000 to 7,000-fold increase in body size during ontogeny in crocodilians. This is among the largest body size increases of any terrestrial vertebrate. Adult American alligators, for example, are 350 kilograms, but they start out life at 45 to 55 grams. And along with this large increase in body mass, we see changes in their cranial form that vary to different degrees with different taxa. So the American alligator undergoes dramatic changes in snout shape and head size. So this animal starts out with a snout that's actually wider than it is long, and a jaw adducter tunnel that's fairly restricted. By the time they get to the adult phenotype, they've become more robust. Their snout has increased in length dramatically, and the pterygoid flanges have moved ventrally and allowed the jaw adductor tunnels to increase in volume relative to the neonate condition. We see similar but not as dramatic changes in the freshwater crocodile, and even sort of fewer changes in the saltwater crocodile. And so I was interested in examining these three taxa as a model to try to see if ontogeny was playing a role in bite force changes that may be significant to understanding their feeding biomechanics and how they exploit various feeding niches. So the experimental design to compare these then was sample bite forces from all three taxa, and to sort of play that childhood game, which one of these is not like the other. So in snout form, the freshwater crocodile has a slender snout, and so if that's driving differences in ontogenetic bite force changes, this should be an outlier relative to the other two taxa. In a phylogenetic context, the American alligator is an alligatorid, whereas the fresh and saltwater crocodiles are crocodiloids, and so these groups share a common ancestor in the late Cretaceous. So if phylogenetic distance plays a role, then the alligator should have a scaling relationship that's different than those of the other two taxa. We measured bite forces again at the same distal tooth position, and we can see there's absolutely no difference in how bite forces scale across ontogeny in these three taxa. These are log just for the span of the data set. And again, body size explains the vast majority of variation, with R-squareds here for all three taxa at 0.97 or higher. So we can say definitively that phylogeny is not playing an effect, and snout form is not having an effect on how bite forces scale in extant crocodilians. And what this points to is a conservation of the jaw adductor musculature, that the gross anatomy of this system hasn't changed over their long evolutionary history, and that's maintained their ability to generate these exceptionally high bite forces. And for those taxa that have been examined by myself and others, that's generally the case. For what's been reported in the literature, we see a lot of similarity in the orientation of fibers and pennation, the muscles that make up the jaw system, where they attach, etc. And that explains at least the performance in terms of biomechanics, but there are important differences between taxa and across development. And so here are the 8 muscles that make up the crocodilian jaw system, and we see these in all extant taxa. In fact, we see all of these in birds as well. So there's a really strong conservation of the jaw adductors and archosaurs generally. But there are differences along the system that I want to explore in terms of physiology. And those are quite dramatic in adults. So here's an American alligator, and here's the false gharial Tomistoma schlegrii, and the same muscles highlighted in both the adductor mandibulae externus medialis. clearly in the American alligator, you can see it's this very deep red color. It's hard to identify in the Temistomene simply because it has this pink to white color that matches all of the surrounding adductor musculature. And so this difference suggests that there's potentially important differences in how muscle physiology impacts their ability to exploit feeding niches, both across their evolutionary history and potentially during development. Now this pattern shows up again and again, so in generalist and blunt-snouted taxa we see there's some amount of red or putatively oxidative musculature, and in the longer rostrum or slender-snouted forms we see this, inverse, that the Temistoma condition shows up in Gavialis gangeticus, and the Mesostops cataphractus is ambiguously reported in the literature, so I don't actually know what's going on there yet, but I wouldn't be surprised to see that it has a condition like Temistoma, because it's also a slender-snouted crocodilian. Now, what's the significance of this? Well, white musculature looks that way because it has low myoglobin content, a paucity of blood vessels, and it relies on an anaerobic metabolism. And then conversely, red muscle looks that way because it has a lot of myoglobin, there's a lot of blood vessels in these muscles, and they rely on an aerobic metabolism. And these differences have potential significance for the way that crocodilians are using their jaw muscles to capture different kinds of prey. So it could be these differences could manifest as potentially differences in force, and we know this from some of the fish literature, that white and red muscle show differences in force generation. But from all of the biomechanical studies that we've done, we don't think that that's the case. Probably there's an insignificant difference in fiber contraction velocity, and that this could be important for taxa that feed on very elusive prey and have to close their jaws very rapidly. This is something that I'm working on now, so I'm not going to talk about that today. But there's also an importance here with the potential for endurance, for the ability of muscles to sustain contraction for long periods of time. And the most substantial difference here is that red muscle can contract up to 5 times longer than white muscle does before it fatigues. And so potentially there's a means that this can manifest in some of the behaviors that especially generalist crocodilians use for capturing large prey. Now this is a video of a Nile crocodile that's secured a zebra, and you might be surprised to know they don't actually try to use their high bite forces to dismember prey once they seize them. In fact, they use them and their high body mass to drag those prey out into deep water. And so how crocodilians dispatch large vertebrates is through drowning them. And they can undergo 20, 30 minutes of engaging their prey before they successfully move it out into deep water. And that suggests that there may be some use for this oxidative musculature to help sustain their bite forces over these long periods of time. And so what I'm proposing here is that the oxidative muscles in their jaw system are mainly what facilitates this prey drowning behavior. And this is what allows crocodilians to be apex predators, is that they take down very large game mammals by this particular behavior, and that behavior may be a result of specifically a transition into having oxidative capacity of their jaw muscles. And we know from a number of vertebrates, including crocodilians, that hatchlings start out with a paucity of oxidative musculature. Especially in the jaw system, hatchlings have very clearly white glycolytic muscles, and that at some point those change over their ontogeny, along with all of these other transformations over body size and snout morphology. Now, neonates start out as insectivorous, and as they move into the adult phenotype, they begin to take on larger and more robust prey. By the time they reach 50 centimeter snout vent length, they've moved from their second to third major dietary transition during their development. And this is the point when they begin to take birds and small mammals. And so these are the kinds of prey that require drowning in order to subjugate. And so I'm suggesting that this is an important dietary transition that should be manifest in changes in muscle physiology if in fact we're seeing a signal from physiology to behavior to exploiting these kinds of prey resources. So then ontogenetically, oxidative musculature should appear prior to the onset of this dietary transition. And so to examine this, I developed a technique during my time at Florida State called Pantone muscle oxidative inference analysis. And this is a rapid way to characterize muscle physiology in in a number of vertebrates or a number of specimens quickly. And the way this works is matching the visual color of the muscle to a Pantone. So Pantone is a specific color pattern that is generated by mixing known quantities of other colors. So here's cyan, magenta, yellow, and black. This particular Pantone 201 PC will always be created when you mix this same combination of colors. And the benefit of this analysis is that we can take magenta as a proxy for redness and yellow as a proxy for whiteness and quantify how magenta or how yellow a muscle is as a stand-in for its relative oxidative or relative glycolytic capacity. And so to do this, I bisected the muscles in the middle and just visually matched the Pantone to muscle color under the same lighting conditions. And this allowed then for identifying gradients or changes in muscle physiology or muscle Pantone as a proxy for that during ontogeny. And what I found is that there are very clear differences in, here's the hatchling condition for all the jaw adductors. Every one of these muscles undergoes a transition to becoming more oxidative during ontogeny, with pseudotemporalis actually developing a core of oxidative muscle early on in development that's not present in the hatchling condition. And when we look at each of these muscles individually, we see this transition. So a single individual here, this is the proportion of magenta and yellow on the Y, and snout vent length on the X. Each individual is represented by a pair of data points, a pink square and a yellow circle. And so at hatching, these animals have a relatively higher amount of yellow in their Pantone, and so presumably a higher amount of glycolytic muscle fibers. At some point, these two curves cross, and they have an even proportion of both magenta and yellow. And in all cases, then the adult condition has a higher average proportion of magenta than yellow, and that's the transition we see to this more phenotypically red musculature. So in this case, then the adult condition has a statistically significant different amount of, or higher amounts of magenta than it does yellow in the adult condition. Now, three of these muscles are very take very dramatic shifts in this way. So adductor mandibular externus medialis here starts off at more than 50% yellow, but by the time we reach the adult phenotype, it's more than 50% magenta. And that's that very deep red muscle that I showed in the dissection photo several slides ago. Adductor mandibular externus profundus does the same thing, and we also see this in the medial portion of the pseudotemporalis. So these three muscles are visually deeply red in color, and we're seeing very strong changes in the proportion of that color over ontogeny, and that suggests that their physiology is changing as they're increasing in body size. For all the other muscles, there were not significant differences between the adult magenta and adult yellow condition, although all of them have an average higher proportion of magenta in the adult phenotype. And so I want to really focus on then these three muscles that become very clearly, quote, oxidative based on this oxidative inference analysis. All of these have inflection points here between 20 and 40 centimeters snout vent length. That's the point when the proportion of magenta and the proportion of yellow are equalized. And then by 50 centimeters SVL, when these animals begin to take birds and small mammals, there's a much higher proportion of magenta in all three of these muscles, suggesting that they are in fact reaching an oxidative phenotype in coordination with their transition into a new ecological feeding niche. And so this then supports the hypothesis that between their first and second dietary transitions, we see a switch in muscle physiology that's concomitant with this ability to now drown prey and access larger, more robust taxa. This same pattern shows up in a cladogenic context or a phylogenetic context. So this is just basically one data point, but Tomistoma schleglii is sort of the taxon that got me thinking about this in the first place. Shows very oxidative musculature based on the muscle oxidative inference analysis. And in its most oxidative muscle is less oxidative than the adult alligator's least oxidative muscle. So there's a very striking difference in this phenotype. And what I actually think is quite remarkable is that the three least glycolytic are also the three least glycolytic in both taxa. And this suggests that there's some potential ability to move up and down, sort of slide the amount of oxidative fibers there are over ontogeny that may correlate to, allow physiology to correlate to the cranial morphotype, and that may actually be the coupling that we thought was going on with bite forces, it could in fact be muscle physiology that's linked to that phenotype. Okay, so back to bite forces. It would be really nice to be able to show that that's actually what they're doing. They're using these oxidative musculature to hold prey. And I think that's what I'm seeing in, this is a real-time bite force plot from a 2-meter-long female alligator mississippiensis. And I'm sure the first thing that you notice is that these animals can regenerate their maximum bite forces over and over again. They can do this for up to 20 minutes at a time. But what I want you to focus on are actually these areas of low force, these plateaus. When these animals engage the bite bar, they won't let go for a really long time. And they're not always generating maximum values, and when they're not, they tend to hold it at about a 10% or less of that maximum force, and we see this over and over again. And what this looks like is you're going to see, so this is a larger individual, it's a male, but it shows the same pattern. I want you to focus on this right here is the ventral pterygoideus muscle. It's going to contract when this animal hits peaks of its maximum bite force. And at this point, the animal's already engaged the bite bar, so we're somewhere in this region where they don't apply a maximum force, but they don't let go of the bite bar either. And so you're going to see that as it clenches down onto the bar, you'll see this muscle contract quite readily. There. And so that was a spike up to a maximum value, and now we're back down into this plateau region. And it's not again, and Greg Erickson's on the other end of this bar, it's not until he starts to try to pry this loose again that the animal re-engages its maximum bite force. There. And then goes back to this low force hold. And so I think this is signal of what these animals do in the wild. That is, they engage their maximum bite forces, and this matches observational data. They engage these bite forces when prey struggles, when they're trying to secure these large and robust prey items. And when, in between periods of prey struggling, they don't seem to engage their maximum bite forces. And in fact, it seems like they're doing something similar here where they may generate these low force holds that allow them to rest in between periods of struggling with their prey. And these are remarkably consistent values. So I've taken the average of these low force holds from a number of individuals. None of them seem to occur before 50 centimeter snout vent length, but they show the same kind of patterns as maximum bite force values do. That is, they're positively allometric throughout ontogeny, and they have a high correlation to body size. And these are from males and females. We see the same pattern in a number of individuals. And across a relative comparison, looking at hold force as a value of the maximum bite force as a percentage of that, they're extraordinarily consistent. So on average, they only range between 8 and about 6% of the maximum bite force. And so all of this together then suggests that we're seeing some intrinsic aspect of this musculoskeletal system that's being activated. And these animals aren't choosing to have low force holds that are highly consistent across a number of individuals. And so what I'm arguing is that the oxidative musculature explains these low force bites exclusively. Now, to get at this, I turned to biomechanical modeling. I dissected a couple dozen American alligators and mathematically modeled how they generated bite forces. And so I used gross dissection techniques that we're all familiar with, but I also used iodine-enhanced X-ray micro-CT. And this is a new method that I've been working on at Stony Brook, and I want to just highlight this, sort of go into some detail about what it is, how it works, and how I used it. Okay, so we know that computed tomography has fundamentally changed the way we do our science. I mean, the amount of anatomy we can visualize using these methods is really quite astounding. And it's given us the ability to do functional analyses, look at phylogenetic characters that are deep with inside specimens, reconstruct soft tissues and fossil taxa. It's been truly remarkable. But there is a problem with computed tomography, and that's that it doesn't image soft tissues. They're not dense enough to create an X-ray shadow. Instead, it's bone, dentin, and enamel that we can image well. And so a number of researchers have been trying to exploit different contrast agents that will allow us to visualize soft tissues in the same way that we visualize hard tissues. And that's what I've been working on with Nate Clay at Stony Brook. And so one of these contrast agents is Lugel's iodine, I2Ki. Now in aqueous solution, I2Ki devolves into an iodine trimer, and this binds differentially to lipids and carbohydrates, and it creates soft tissues that are effectively as dense or more dense than bones. They become radio-opaque, and in an X-ray micro-CT, they are can be easily imaged. This has been explored in the developmental literature for the last several years. So here's your standard set of sort of evo-devo taxa, a mouse model, a frog model, a bird model, and a fish. And you can see that in these developmental models that the organs of the thorax and abdomen are quite readily visible. Specific organs within the eyes of this fish are quite remarkable. And comparative anatomists in the last 2 years or so have started pushing this to try to get it to work on larger and larger specimens, on post-embryonics. Now for neonatal specimens, it works quite well because they're small. But as we get into larger individuals, like adult mice here, or this yearling American alligator, we're getting problems of coronal effects, that the stain is only really working on the periphery of the specimens, and that it doesn't penetrate deeply, and so these specimens aren't fully visualized using X-ray methodologies. Well, Nate Clay and I have been exploring ways to improve this, and here's what we've come up with. This is a hatchling American alligator with slices moving from the palate dorsally through the head, and what you're seeing is just about every soft tissue in this animal. Be they jaw musculature, cranial nerves. Within the brain, we're identifying both myelinated and non-myelinated regions, so there are processing centers there that we can visualize. Orbital and cranial fat pads and glands. Here the lens is showing up as a highly glucose-based metabolism, so it picks up a lot of iodine. And so from these kinds of images, we can basically do all the things we do with hard tissues to soft tissues, and really get a great sense of three-dimensional anatomy, and especially small and delicate specimens. And so how we've done this, we've used immediately euthanized yearling and hatchling American alligators, and this is just so we have the best anatomy to work with. We stain them with an 11.25% solution of Lugel's iodine. Basically, we're just submerging them in iodine and leaving them there for a few weeks. If you can marinate a steak, then you can use this method. These values are up to 40% higher than what we see people have worked on for embryonic specimens, and we think that's particularly important for getting it to work in larger individuals. We use a 210G Phoenix VTOME high-resolution micro-CT. All of our data sets are between 30 and 40 microns, and our beam energies are very high, 150 to 200 kV. These are the kinds of beam energies you'd use for differentiating fossils from surrounding rock matrix. And they're up to 9 times higher than had been used for embryonic vertebrates previously. And it's this combination of really extreme staining, concentration, and very high beam energies that's giving us this great visualization. And so what we're able to differentiate then are sort of the classic butterfly of spinal white and gray matter. You can see here the jaw adductor muscles, individual muscle fascicles are visible. We can see very small details such as alveolar nerves or the lateral palatal nerves in the palatal mucosa. And so to say that we're very excited about this would be an extreme understatement. We are exploring a lot of ways to sort of enhance this method to get at larger and larger specimens. We've recently had it work successfully with birds. This is a 14-day emu, and so we're getting similar kinds of results. And just the week before, just last week, we got it to work on an adult emu. So we're now moving into potentially full ontogenetic series where we can image three-dimensionally the soft tissues to the same degree we can the hard tissues of these animals. What do we do with them? We reconstruct the anatomy. So here is a hatchling American alligator. A series of extrinsic ocular muscles are reconstructed in sort of this salmon color, the brains in blue, and then in this case the nerves passing to the orbit are all identified. So the trochlear nerve, the oculomotor, abducens, and this extraordinarily large trigeminal nerve. And then we can use these to correlate a number of, well, basically anything. We can look at how form and function in these soft tissues relates to performance, which is something I'm working on with a high school intern, as well as examine ways to reconstruct soft tissues in fossil taxa, etc. So we're extremely excited about this. What I'm using it for here is identifying parameters of muscles that can be useful for biomechanical analysis. So in this case, I've highlighted the adductor mandibulae externus profundus and then an individual fascicle. And from that, we can then volumize the muscle, identify muscle fascicle length, use that to determine physiological cross-sectional area, get the orientation of the muscle in three-dimensional space, determine its pennation angles and where it attaches, and basically determine how much force this muscle can put out in a biomechanical model. And so that's what I've done with my smallest specimens in this, because they're difficult to work with, and this really enhances sort of the precision in getting at their modeling their bite forces. And there's one other thing we're going to be exploring this summer about this is, and it's something I still find remarkable. you probably notice in this blown up adductor tunnel series that there's inconsistent staining. Some of these muscles are very white, some are very gray. And we've seen this and we thought it was a problem of incomplete staining, but it shows up on the left and right sides. It shows up in multiple individuals. And I want to bring your attention to this gray area here. What I think we're seeing is signal of muscle physiology. So this area highlighted here is this same oxidative muscle in an adult American alligator. And because iodine binds to carbohydrates, and oxidative muscles have a lower carbohydrate load because they have a different metabolism than the glycolytic muscles around them, they shouldn't pick up as much iodine, and they should be grayer in our contrast-enhanced CT images. So I think that this is potentially giving us some really strong signal about what physiology of muscles are, and it's giving it to us in 3 dimensions for an entire individual for all the muscles that we're staining. It's something that I'm going to be exploring thoroughly this summer, linking the muscle oxidative inference analysis with this and doing immunohistochemistry to try to match that up. So invite me back in a couple of years and hopefully I'll have all of that figured out. Okay, so then for the biomechanical modeling, for large specimens I used gross anatomical dissection, but for these smaller ones I used iodine-enhanced micro-CT. Now I was told there's this phenomenon called Berkeley time on campus. So I trimmed my talk down a little bit to account for Berkeley time. So this is where there's supposed to be a lot of methodology about how I developed my model. If you're interested in that, I have the slides after the acknowledgements. I'd be happy to go through them. But I'm just going to summarize that, and if you want to ask about it during question and answer, that's totally fine. Okay, so the biomechanical model makes some standard assumptions that the skull's a rigid body with a frictionless pin hinge. There's recruitment data for jaw muscles available in the literature for Cayman crocodilus, so I'm assuming that those data are comparable to what alligator would do. I assume that the right and left jaw adductors contract the same way, and I use an average muscle stress across all the muscles and across ontogeny of 33 newtons per square centimeter. This data comes from Dipsosaurus dorsalis from a study in 1984 by Johnson and Gleason. I've done my own whole muscle stimulation on alligator, and I get a value of 32.4 newtons per square centimeter, so it's basically comparable to this. But this data set, theirs is more robust than mine is at this point, so I'm still using these numbers in my model. As I develop my data more, I'll switch it out. At this point, I have one individual of alligator, and I don't want to assume that that individual in that one muscle is representative of everything in the taxon. And so here is the biomechanical model. Right and left sides are accounted for by doubling the muscle moments for all the jaw adductors, which are summed together. One muscle inserts both anterior and posterior to the jaw joint, so I have it 2 moments for that. And then the ventral pterygoideus muscle, this is this guy, which sits out here on the retroarticular process, is highly pennate. So I use Scott and Winter's variable pennation angle equation to account for how fiber angles change as the muscle goes into full contraction. And so that allows me to not overestimate how much force that muscle generates. And then I'm particularly interested in forces that are generated at the most prominent canine. This is the tooth they use for seizing prey, and so then applying this model to these hold forces is going to be relevant for getting a model that is correct for this bite point. Okay, so I'm testing this model. These are maximum values against in vivo measured bite forces from an additional population of American alligators, and the model data I'll overlay in blue. You can see that it's not significantly different at all from in vivo data. So I'm extremely happy with how this model's come out, and what we can do now is go into the parameters of the model and change them to model a different kind of bite. In this case, to model a low force hold that I'm arguing is generated by these oxidative muscles and that facilitates this prey drowning behavior. Okay, so for hold force generation then, I'm interested in these oxidative set, this particular 3 set of muscles that should be facilitating this behavior. So I only want to model those, but I do have to account for one other muscle, and that's the intramandibularis. And the reason for that is there's a cartilago transillians, this little cartilage sesamoid, that inserts on the dorsal part of the, or into it dorsally insert the adductor mandibulae externus profundus and the pseudotemporalis, and ventrally the intramandibularis inserts onto it. So to mathematically balance the model, I have to include this muscle, but I only allow it to generate enough force to equalize the dorsal force generated by these 2 muscles. And so the hold force model is much simpler, but it's the same idea. Right and left sides are accounted for. The intramandibularis is accounted for by doubling the forces of those abductor mandibulae, externus profundus, and pseudotemporalis. And then I'm modeling what the bite force would be out here at this most prominent canine, since this is where these animals typically seize prey and begin to generate this whole behavior. Okay, and so the model data are again in blue, plotted alongside the in vivo data. You can see that the two data sets are not significantly different. In fact, they're both positively allometric. Isometry here would be a scaling coefficient of 2.0. And they're both highly correlated to body size. So it looks like then that these oxidative muscles are what's controlling this low force hold in adult American alligators. And taking this data as a relative comparison as a percentage of the maximum hold force, or hold force as a percentage of the maximum bite force, we see the same pattern. So where the in vivo data falls between 8 and 6% of the maximum bite force, the model data falls between 7 and 5%. The two data sets aren't significantly different. Now, I sort of expected that this would happen, but really this could have a slope of 0 that just spanned across this range, and it would be statistically not different. That the slopes and the scaling relationships of the model and the in vivo data are so very similar suggests to me that the model really is accurately accounting for the physiology and therefore the behavior of American alligators in generating these low force holds. Okay, so then these putatively oxidative muscles in the adult condition fully explain hold force capacity. So what is this telling us? What's the narrative so far in terms of how crocodilians are so very successful at being apex predators in these shoreline environments? Well, these long-held ideas that cranial morphology highly correlates to bite forces are completely wrong. So we'll get rid of those. But it seems like what we're seeing is a strong relationship between muscle physiology and cranial form. That's the signal, that's the coupling of this complex phenotype. And that behavior, or that physiology facilitates a key behavior that allows these animals to occupy niches, at least generalist crocodilians, to occupy niches as apex predators. What's more, the inverse is also true. So in taxa that don't feed this way, that don't drown their prey, neonate alligators and these fish specialist temistomines, we see that they have highly glycolytic muscles. And what this suggests is that there may be a heterochronic mechanism where neonates generally are more like the adult temistoma phenotype regardless of taxon, and that could allow them to evolve across, evolve differences in physiology as they're invading new niche space at cladogenic events. I think that, well, where I want to go next with this is looking at velocity, jaw closing velocity. And part of the impetus for this is this last portion where glycolytic musculature is prevalent in neonates and in longer rostrum crocs. And part of this in the American alligator is this really terrible design of a snout. These animals hatch as jaw prehension feeders, but they've got a jaw that's wider than it is long. It's really a pitiful kind of tool for catching prey. But having highly glycolytic jaw muscles may facilitate faster jaw closing velocity, because we know that these kinds of fibers are faster contracting. And so this may explain why hatchlings in this case have this very glycolytic phenotype. And then the same can be argued for slender-snouted crocs. So looking at I'm doing experiments with high-speed video to look at jaw closing velocity in subaquarial and subaquatic, subaerial and subaquatic environments. And the same should hold here, that these are animals that are capturing elusive prey, and so having highly glycolytic jaw muscles with faster contracting fibers would also help them in enhancing their jaw closing velocity. So I think that what's going to happen here is that there's not going to be a dichotomy between physiology and cranial form so much as different aspects of physiology are going to facilitate different behaviors, and that's how these animals are moving into new feeding niches, both ontogenetically and across cladogenic events. This was a lot of work, and I basically could have been done with an army of people to help and advise me, as well as a number of organizations for funding. And I'd like to thank all of them, and thank you for your attention, and I'd be happy to take any questions.
So some people may need to jump up and run out to go to their next class, so we'll give people a moment to do that, and then we'll let Paul field his own questions. Okay, let's go ahead. start with the questions. I think Dave's anxious.
I'm certainly very interested in your imaging. That's really impressive. But my question is about the ontogeny of the muscle coloration that you did. How did you arrange that? How were you able to look at the color of the muscle, if I understand it correctly, in the same animal through life?
No, so this was a developmental series of individuals for which we had bite force data, and then were sacrificed, and then I examined each muscle in those individuals.
And you had to do that with freshly? Immediately
sacrifice. So we work with local nuisance trappers, and so we know, so we'll measure the bite forces of animals they bring in. They keep them alive for about a week or so before they begin processing, and as soon as they do, we go and get the heads, and so we can put all of those data back together.
I see.
So first I'm going to complain because now I have to change my herpetology lectures. Because I like to talk about the correlation of the skull morphology of crocodilians and bite force.
Doesn't happen.
Yeah, which isn't true. I mean, that was really great. I mean, it's totally believable as well. I mean, I guess the part of it that, the side of the argument that I've always thought intriguing, in part because they're analogous systems, is fast jaw in Gavialis and such, because their phenotype is so similar to that of, say, river dolphins and things like that. And I wonder if there's any literature that's been done on the whales, actually, or any other sort of analogous systems that might give you any insights into how that's working, and whether it's really speed or whether or not it's just being more hydrodynamic, as if they're doing side-to-side movements and things like that.
There is some work on whales, and it's actually this part of it's here as it's closing its jaw. This wave is analogous to what we see in whales as they're, especially baleen whales, as they're gulping in large amounts of water. And so there's some interest in what the material properties are, how elastic this region is, and whether or not it contributes to basically dropping so that it reduces the bow wave created by the jaws as they're closing. So yeah, there are analogies there, at least in this regard. But into the musculature and behavior, that we haven't explored yet.
So can you do your imaging with long dead material, or does it have to be freshly
It will only look as good as the tissues look anyway. So specimens that are preserved will work. It does not work well with alcohol, so it needs to be, iodine in an aqueous solution is needed to osmotically move into the specimen. And so, yeah, and so we fix them in formalin. And so if you have an alcohol specimen, you fix it in formalin, push out the alcohol, then it should be fine for using.
Okay.
Exactly.
That's what Walter Bach advocated a long time ago.
Okay. Now, as to whether or not something like the physiology signal shows up in an older specimen, I will tend to doubt it. Simply as you process and put more chemicals through tissues, that should wash out some of the components, so the signal may not be as strong.
Right. It's certainly beautiful.
Yeah, for sure. Should there be a biochemical signal as well? Like, is there some other type of genetic analysis that you could do to try to get a handle on those the difference in the muscle?
I'm not familiar enough to really know how to tackle that.
I think what you've done is pretty compelling.
What about lactases?
Yeah, so that could be an avenue. Presumably we should see differences in how much waste output that the muscles are generating. But I think, at least for comparative anatomy, taking the immunohistochemistry route is sort of the more standard way to approach it. And so that's, at least our first step is going to be examining it that way. Sarah?
Does the iodine do anything to the specimen? Because, I mean, just thinking about Dave's and Marbley's comments about using preserved specimens, it seems like if the iodine doesn't damage the specimen, you could force the alcohol out, and then you might have a really good shot at doing the stuff that we normally would want to dissect out and put on specimens that taxa for which we only have one. I'm thinking about all of those holotypes, Marbley, of Sicilians that we only have one of. So it just seems or whatever taxon we only have one or two of.
Well, I can say that Lugol's washes out pretty readily.
A sodium thiosulfate solution will wash it out fairly immediately, actually. So you can use museum specimens and then wash them clean, and no one will know the difference.
Right, it seems like the combination, because you get the I mean, it looks like the bones are showing up in these scans just as well, so it seems like this method could kind of be a one-stop shopping for...
It could be. You do get bone. The iodine tissues are denser than bone, so the bone shows up as a lighter gray color, though it's there. The technique we've been using is to scan specimens for hard tissues first, then stain them, and then we can fuse those two data sets to give us everything that we need. There is an effect of the iodine, so we see, this is a time series. What we see is that things like the ventricles get larger over time, so we're seeing some shrinkage in the specimens. That's because they're basically putting a salt gradient in them that's drawing the water out, and so accounting for that, you then use that in your analyses. This is how much volume changes there are, and so then you can sort of correct for that issue. That's the only side effect that we've observed.
How long does it take to do one of these scans, just out of interest?
So at 11.25%, we did a time series in order to test the optimal staining period. So at only a week in a hatchling American alligator, you don't get full penetration of the iodine. And 4 weeks plus, there's so much iodine that it begins to get attenuation in the scan. So our sweet spot is 2 weeks for hatchlings, 4 weeks for yearlings, and for hatchling emus, and 15 to 16 weeks for adult emus.
Oh, wow.
Do you think that if you injected the iodine, you could speed it up?
I think you could. I would worry that you would get an artifact of the injection at the point you injected it. So if you were going to do it, I would say you should do it through a vascular system that allows the iodine to move readily, and know that where the artifact will be is in a known anatomical point, preferably away from a particular region of interest.
Right, you could do it into the jugular.
Yeah, exactly. We're kind of playing around with ways to modify this for different kinds of systems, but we're also just interested in giving it out to the world to see what you all do with it. All the ways that you can apply it to your systems and sort of make it a better technique, for the community in general.
Regrettably, we don't have a CT operation right here. What does it take in your estimation these days? Does it take not only the machine but a technician?
Yes. Well, you need someone to run the machine, I guess.
Yeah.
But that's, we train students to do that.
Well, that's the point. Can you train? Does it take? You can.
Yep. So the scanner that we use at the AMH is an industrial strength scanner. It's designed for automotive components and car parts, which is why this is working on a scale that we haven't seen other labs produce, because biology tends to have lower powered micro CT scanners. This one's used for fossils, and so we're basically giving it a super dense set of tissues, and it's able to resolve that really well.
But I hear from the students there that the machine is down
Constantly.
half the time.
Yeah, I lost a scan date last month because of that. It's being used 12 hours a day inside design for that.
More questions for Paul? Well, those of us who are still here, let's thank him for excellent talk.